Good evening and thanks for attending. For those of you who don't know me, my name is Rich Llewellyn. I'm one of the assistant chiefs here at Everett Fire. We're excited to be able to offer this educational opportunity tonight. And I know we also have Julie Sartosky here, and she will also get an opportunity to introduce herself. She's been our liaison with Fred Hutch since we began our COVID antibody study with Everett Fire last year. So we'll let her introduce herself as well.
Okay, so we're going to mute ourselves and step away. Dr. Shapiro, Julie, if you'd like to take a minute and introduce yourselves and then go ahead and get started. Thank you.
That's so great. Julie, since you are the liaison and the known entity with this team, why don't you go ahead and introduce yourself first, and then I'll give my intro and jump into the presentation.
Great, that sounds good. Thanks, Adrienne, for being willing to do this. I can't see everybody, so I don't know how many of you may have met me. We've been working with the Everett Fire Department, looking at if there was infection in folks that had been exposed early on, and then those that have been infected following their antibodies over time, and those that haven't, also following them over time. And it's just been a real pleasure and a real honor to work with this group. And so take it away, Adrienne.
Great. Thanks so much, Julie. Julie is a nurse practitioner and researcher at the Fred Hutch, the vaccine trials unit, and is the lead clinical investigator on the COVID cohort study that some of you may be a part of, seen the team around. It has been a pleasure for me to work with Julie and to meet your department and the Snohomish County Department folks who are here. So thank you so much for having me. I'm Adrienne Shapiro. I am an infectious diseases specialist doctor. So I see patients in clinic and in hospital with folks who have infectious diseases. And I spend some of my time doing clinical work and some of my time doing research. So in addition to my clinical infectious diseases specialty, I also have a PhD in epidemiology. And the research that I do when I'm not seeing patients includes virology, immunology, bacteriology, and epidemiology. So all of the ologies. And it's a real privilege to be able to talk with all of you about things that I, like you, have spent the last year and a half working on, just in different ways. In addition to the research and the infectious diseases specialty work I do, I'm also a primary care doc in Everett. So it's particularly nice for me to be able to talk to Snohomish County folks. I see patients at Snohomish CHC at the Everett South location, and so you guys you folks are really my home territory. So I'm really glad to get this chance to talk with you. We're going to go through some science. We're going to go through some clinical questions. And I have these conversations with my patients every day that I'm in clinic. I'm so delighted to have them with you. And I really welcome questions. Let's try to keep questions during the presentations to questions that are really focused on what I'm talking about. And then if you have questions that I don't cover on topics I don't cover in the presentation, let's save those till the end. But I want to make sure what I'm saying is clear and makes sense during the presentation. So please feel free to put your hand up during the presentation. So your chief was kind enough to ask you all to submit some questions that you had, and went through the questions and figured out what are the top 10 questions that came up among the responses that came from the departments. So I got the list of the top 10 questions, and the presentation is designed to hit on all of these. And the way I saw it, looking at the presentation, they kind of fell into 4 buckets. So the questions really addressed 4 key themes of what are the vaccines? What are they made of? How are they made? Where do they come from? So we're going to talk about that. Then some really key questions, really important questions about, are the vaccines safe? What are the risks of the vaccines? We will absolutely talk about that. Some questions that really kind of got to, why are we even doing this? Why are we doing the vaccines? What are the vaccines? How are they going to address the COVID-19 pandemic that has taken over our lives? And then I was so gratified to see that some questions really fell into the bucket of, tell me more about infectious diseases and epidemiology. These are things I love to think about, love to talk about, and happy to share my thoughts that were conveyed. So these were how the questions kind of fell into the different buckets. I won't read through each one of them, but let's just dive right in to bucket number one. What are the vaccines? What are they made of? What's the difference between the 3 vaccines? And just so we're all clear for purposes of this discussion, we're going to be talking about the 3 vaccines that are that have emergency use approval or FDA approval in the US. So that's the Pfizer and the Moderna vaccine, which are the mRNA vaccines, and the Johnson & Johnson vaccine, which is the an adenovirus vaccine. So one question that seemed to come up was, when were the mRNA vaccines developed? A lot happened in the last year, but the mRNA vaccine development process has been going on for decades. So this is just a table that comes from a journal article in 2018. So before there was any knowledge or any twinkle in any bat's eye that COVID could be an issue, this table lists all of the different mRNA viruses that were mRNA vaccines that were in human trials. So these were not for COVID, obviously, because COVID didn't exist yet, as far as we knew. But there were mRNA vaccines that have been developed and were in phase 1 and 2 trials for HIV, for rabies, for Zika, and influenza. And I'll just call folks' attention to the bottom of the screen here. Moderna, the company Moderna, has been working on mRNA vaccines for years. COVID's not their first mRNA rodeo. So they had Zika virus and influenza virus mRNA vaccines in trials back in 2018. And then this is a very busy slide. I'm sorry about that, but it just shows how far back the timeline goes for the development of mRNA vaccine. mRNA was discovered in 1961. And then by about 2001, there were the first trials of studies using mRNA technology in preventive or therapeutic efforts. So going back to 2001, there have been, and then maybe 2009 was the first mRNA clinical trial of a formulation. 2017 was the first in human test of mRNA vaccines looking at cancer. So just to say, these timelines show mRNA vaccines, the technology and the research, the foundations go back a long time, so that a lot of the platform was ready to go when COVID hit, and the genome was rapidly sequenced in early 2020. And so they were able to plug in the key elements into a well-researched platform to develop the mRNA vaccines. So these mRNA vaccines that have been developed for 20 odd years, what are they and how do they work? So I'm going to call your attention to start to the graphic on the left side of the screen here. So fundamentally, mRNA vaccines use pieces of genetic material of the virus that give the blueprint instructions to make a piece of the virus. So mRNA contains the blueprint for making the spike protein of the coronavirus. The vaccine is given into the muscle, in the arm. Then that piece of mRNA, that piece of genetic material, which is shown here in another diagram, it's covered in what's called a lipid nanoparticle, which is basically fat cells that helps it get into cells in your body. And then that mRNA comes into a cell in your body, and your body's cell machinery takes that mRNA, reads the blueprint to make spike protein that belongs to coronavirus. But in this case, it's just the protein, it's not the rest of the virus, so it can't cause any harm. Once the virus enters the cell, the human cell mechanism makes the protein and then displays that bit of protein on the outside of the cell so that the immune system can see it, can learn to recognize it, and form defenses against it. That way, when the actual virus encounters the immune system, it's all ready to go with its defenses. That I should also just mention that the mRNA that gets introduced is destroyed within about 24 hours. So it doesn't hang around in the body. What hangs around is not even the spike proteins, because those get made, but then they also get destroyed. What hangs around is the immune response, and the vaccine itself disappears within hours to days. There's no traces of what was originally there. So that's the mRNA vaccine, and that's the way both the Moderna and the Pfizer vaccines work, that same principle. The Johnson & Johnson vaccine is a little bit different. Instead of using a piece of mRNA surrounded by fat cells to get into the body, Johnson & Johnson vaccine takes a piece of DNA, a different kind of genetic material from the coronavirus, puts it inside a weakened adenovirus, so a virus that causes the common cold, but has been engineered that it can't make people sick and it can't even replicate. So once it's dead, it can't make copies of itself to cause more copies, and it can't make you sick. So it really is just a vector. It's just a transportation mode. So it carries the adenovirus, which is this yellow spiky thing, carries the DNA into a human cell. The viral vector then sort of injects the DNA into the human cell. The DNA is turned into mRNA, and then we're kind of back where we would be with an mRNA vaccine. So it all gets to the same point of our bodies reading that blueprint to make spike proteins to train the immune system. But the adenoviral vector, the Johnson & Johnson, is delivered through an adenovirus, whereas the mRNA vaccine is delivered just in those fat particles as part of the vaccine. Other key differences between the vaccines: Johnson & Johnson is delivered as a single shot, whereas the mRNA vaccines are both delivered as a series of 2 shots as a primary series, with 3 to 4 weeks in between. On a practical level, the Pfizer vaccine is available for people aged 12 and up, whereas Moderna and Johnson & Johnson are for aged 16 to 18 and up. And there's some differences in how cold the storage needs to be to transport them, but functionally they all get you to the same place, which is having your body produce spike proteins to then train the immune system to recognize the invader, which is COVID, when you recognize COVID in real life. And then there's some minor differences in efficacy, in how well each of them works. It's looking like the Moderna vaccine probably has the longest acting efficacy of all to prevent serious infection, serious disease. Pfizer's, but they're all very good. They're all in the high 80s to 90s of preventing severe infection and hospitalization and death. They're all excellent, in fact, at preventing death. So importantly, none of them is 100% protective against getting infected in the first place, but because their job is to train the immune system to quickly respond and take down the virus once it gets into people, they all do a very, very good job of preventing an infection from causing illness. They also all do a good job of preventing infection, but the better, the more reliable efficacy is preventing severe disease. All right, and then this is just a quick slide to highlight the fact that in the before times, before 2020, the development of a vaccine process took probably on the order of 10 years, in part because everything was done in sequence. So the basic research started, and then trials in people, phase 1, phase 2, phase 3, and then the vaccine would be licensed, and then it would be manufactured, and then it would be delivered. All of that entire process, every single one of those steps was done in developing the COVID vaccines we now have. It was compressed in a timeline because many of those steps were done simultaneously. So they started manufacturing vaccines before we knew which ones were going to work. It was taking a gamble, and with the hope that of the many that were going into trials, at least one would work out, and then that would be, or those would be the ones that go forward. So that timeline was able to be compressed to less than a year, in part because a lot of the preclinical development, early lab science had been going on already, and it was easy to convert vaccine research for other diseases into vaccines for COVID, and then compress that timeline with the support of a lot of scientists. So some folks had questions about what's in the vaccines, and specifically some folks were concerned about whether fetal cells from aborted fetuses are used in COVID-19 vaccines. So I want to be very clear. I've got a table here showing each of the vaccines, and none of the vaccines contain in themselves, in the product that goes into your arm, none of them contains any fetal tissue from aborted fetuses or any kind of fetal tissue at all. All of the vaccines at some point in the development, usually in the very earliest research testing part, not involving any testing in humans, some did use fetal cell lines in the very early stages. So in fact, all of them had fetal cell lines used in the very early stages of development. And then there are fetal cell lines that are used in vaccine production for the Johnson & Johnson vaccine, not for Moderna and Pfizer. And I want to be really clear with what I'm saying. What is a cell line versus what is a fetal cell? So these cell lines are developed, the ones that are used in Johnson & Johnson, for example, the cells were taken from a fetus in 1985 in the case of the cells used in Johnson & Johnson. The cells were taken from a fetus, they were multiplied and replicated in a lab over generations and generations of the cells. And that turned into a cell line. So none of those cells are really the original cells that came from the fetus, but they've been replicated and multiplied over cell division generations. And then that cell line is used in research and development, in the case of Johnson & Johnson, in production. So now in 2020, the generations, thousands of generations of descendants of those cells are used to make basically the virus that is used in the Johnson & Johnson vaccine that grows well in these cells. From Moderna and Pfizer, there's no part of fetal cell lines or any fetal products that are used in manufacturing the vaccine itself. The fetal cell lines were only used in the earliest phases of testing and safety to understand what kind of effect it has, the vaccines have on human cells. And for comparison, that same kind of early laboratory testing that's done on Moderna and Pfizer with fetal cell lines, those fetal cell lines are used to test almost every prescribed medication or over-the-counter medication that we have. So that same kind of testing process that was used for Moderna and Pfizer was used for Tylenol, aspirin, Lipitor, remdesivir, which is a treatment for COVID, ivermectin, which is not a treatment for COVID, metformin, lidocaine, Prilosec. Pretty much any medication you can think of has been tested on fetal cell lines. So that's the degree to which it's involved. And the Pope, the Catholic Church, and many other religious organizations who are all very opposed to abortion have all given license to for congregants and observant folks to receive the vaccine, given the overwhelming balance of necessity and be able to protect humanity that comes from that. I hope that offers some reassurance to folks who are concerned about aborted cell lines. So safety. Bottom line is the vaccines are incredibly safe. They are not 100% safe because nothing is 100% safe. The Top Pot donuts that I brought into clinic today, not 100% safe. In terms of what we know about vaccine safety, over 1 billion people have gotten the vaccines. They've been extensively tested in hundreds of thousands of folks in the US who have participated in clinical trials. We know that there are, with all of the vaccines, there are common mild short-term side effects right after getting a vaccine. So things like arm soreness, maybe some mild fever, chills, feeling kind of crummy for a day or two, but then those go away. So they're short-term, they go away, quite mild, and those are common. Almost everyone who gets a vaccine experiences some side effect of the mild variety. There are some extremely rare and very serious side effects as well. And these are side effects that occur within hours of getting the vaccine, up to about 8 weeks after is the longest you would expect to see any of the serious side effects occur. And these serious side effects are things like allergic reactions, anaphylactic reactions. The J&J vaccine is associated with a rare type of a blood clot, which can be quite serious. And the mRNA vaccines may lead to an increased risk of a certain kind of heart inflammation, inflammation of the lining of the heart or the heart muscle itself. I should say that even the most serious of these, the blood clots and the heart inflammation, are rarely fatal, especially now that we know that it's a possibility that they may occur. We have the ability to recognize and treat them appropriately if they were to occur. The vast majority of people who experience heart inflammation recover from that heart inflammation with no long-term sequelae from that. So it is not there have been one or two, I don't want to say one or two, there have been small single-digit numbers of people who have died from these rare and serious side effects. But compared to the literally billions of people who have received the vaccine, it's much safer than getting into a car. And I also want to reassure folks that there are no long-term side effects. It's actually not even a thing that happens with vaccines. So if a side effect is going to happen, it's going to happen within the first 8 weeks. No vaccine in the history of vaccines has ever had a side effect show up 2 years, 3 years after getting the vaccine, and have that be the first time it shows up. So if something happens, you're going to know about it in the first 2 months. And just for perspective, when I say rare serious side effects, these heart inflammation side effects, it's about 12 cases of that happening per million vaccines. So really, incredibly rare. And the comparison, and I think this gets to one of someone's specific question is, what's kind of the trade-off of the risk of the vaccine versus the risk of COVID? COVID is incredibly dangerous. You folks have really been at the front lines. I don't need to tell you that people get incredibly sick. Many people get very sick. COVID itself causes blood clots and heart inflammation at 20 to 30 times the rate that what you would see with the risk from a vaccine. So you really don't want COVID. People need to be hospitalized. There is long-term permanent organ damage and disability that comes from COVID. And even for the many folks who have relatively mild cases of COVID, they can still get long COVID, which is prolonged symptoms, often shortness of breath or kind of a brain fog, really a wide variety of long-term symptoms that don't go away for months. There are some folks who have long COVID and we haven't seen them get better yet. So the risks of COVID are real and profound, and on balance, it is a much, much, much greater risk of death or disability from COVID than from the vaccine. So it's really not an equivalent trade-off. Vaccines are overall very, very safe, and COVID, even if COVID doesn't put you in the hospital or if it doesn't kill you, it can make you sick and it can give you long-term permanent damage from even a mild illness. And we see that even more so in younger folks, in fact. So then to some questions of, what are we doing with vaccines? You've all had PPE. Isn't PPE enough? And why should you get vaccinated if you've had COVID and have immunity from COVID? So I think my brief answer is that PPE, it's good. You should use it. If that's all you've got, you should really, really use it. But vaccines give you an extra layer of protection that can't be guaranteed with PPE alone. I work in Harborview Hospital in Seattle sometimes, and we've seen cases of hospital staff who are always masked, always wearing PPE, ending up getting COVID from a patient who was not masked at one point in the room. So PPE is great. It prevents a lot, but it's not perfect. And you wearing PPE does not guarantee that other people are going to wear PPE. And if you're responding in an emergent situation, you have no control over what the folks you're going to encounter are wearing and using, if they're doing it properly. And so having that extra protection from the vaccine is key. It's your long-term insurance policy, whereas the PPE is sort of a short-term umbrella. Your flood insurance, where the PPE is an umbrella that's probably just going to blow out in Seattle winds anyway. And then I think I'm really glad somebody asked the question of why should you get vaccinated if you've had COVID and have immunity from COVID? And that's the answer to that question. We're really very indebted to the Everett Fire Department folks who were part of the study that Julie Sartosky led, that showed how immunity works from COVID infection compared to COVID vaccination, and which combinations lead to the best degree of protection. So this slide is taken from a colleague of mine who did a similar kind of town hall. And she pointed out that the immune response from an infection is really not predictable. Some people do develop very good protection, but many folks don't. And the protection that you get from a COVID infection may go away at some point, and with timing that we don't really know. And there's no good way to check. You don't want to be sending off blood to a lab every week saying, do I still have it? Do I still have it? Am I still protected? We don't know how that's going to work. And the other key point of that is that the immunity you get from an infection is going to be much more specific to that infection and less general to protect against the variants than what you see with the vaccines, which protect better against the variants of concern, including Delta. This is a little bit of a frightening slide. I have to admit, when I first saw it, Julie sent this to me. I got a little concerned, but I'm going to walk you through it because this is the data from folks in Seattle, Everett, King County, Snohomish County, Pierce County, who participated in a study. And Julie and her group and our lab colleagues asked the question. So these dots show the levels of antibodies and the amount of antibodies that reduce the infection if in a lab they're exposed to COVID. So these first four graphs here show the level of immunity, so the level of antibodies that folks had after an initial COVID infection. So there's some, and you can see these clusters of squares show decent levels at the kind of 100 level of titers of antibodies. And then this is what happens, this next set of four colors is what happens after those folks who were infected got one dose of an mRNA vaccine. And you just see the titers shoot up. I want to point out the scale. This isn't a 1, 2, 3, 4 scale. This is going up by powers of 10. So you're going from levels of kind of average around 100 after vaccination up into the 10,000s. Levels around 100 after infection to up around 10,000 after one shot of an mRNA vaccine. So this is kind of a superpower. You really boost your immunity and your antibody levels if you get vaccinated after a COVID infection. This is what happens on the right side is the graph that shows what happens to people's antibodies, people who were not infected and then get vaccinated. And you can see that folks who are not infected, they have no antibodies, understandable. And then they get 2 doses of an mRNA vaccine and the antibody levels jump up. And they jump up a lot, but it's still not quite as high. So really, if you were infected and then you get vaccinated, you've kind of won the immunity lottery, because the highest immunity we're seeing is in fact in people who were infected and then get vaccinated. You obviously, the ideal would be to not be infected at all, but if you are infected, capitalize on that by boosting your immunity even further with vaccination. All right, so I think I've covered most of the key COVID-19 infection and vaccination content questions. And now I'm going to go to the IB, infectious disease and epidemiology bonus round, the questions that ask some about the vaccines, but really ask about some infectious disease questions in general. So one question was, and I love this takes me back to my epidemiology PhD days of working out these questions in lab and in lecture. So one question was, if all of the COVID-19 cases that are reported are only measured by the number who tested positive, doesn't that mean that there are a lot of undetected cases, people who never tested, in which case the case fatality rate and the case hospitalization rate are actually much lower? So this is a really astute question. That is absolutely true. So the denominator, the true number of cases, we don't have that measured, because we don't have that reported, because many people who have COVID don't get tested. So their numbers can't get reported. So that's the denominator of cases. What we do know, what we have really good numbers are the numerator, the number of people in hospitals and the number of people who die. So it is true that of the observed reported cases, the true case fatality rate, the true case hospitalization rate is lower, because the true denominator is bigger than what we see, than what is recorded. But the estimations that we make of case fatality rate and case hospitalization rate don't just come from reported cases. It also comes from surveillance testing, where we go out and we test people who are asymptomatic. We test folks who don't want to get tested, who weren't planning on getting tested by themselves. And that gives us a sense of what is the true population prevalence of COVID, who's actually getting COVID. And then we can use the numerator that we see of people coming into hospitals and people dying to more accurately estimate the case fatality rate and case hospitalization rate. So through a combination of some modeling and some surveillance testing, we do get pretty accurate estimates of how serious things are. But whoever asked this question is 100% correct that many people, particularly people with no symptoms, who don't know if they had an exposure, are not getting tested. And we see that when we look at blood donation data, we look and see who's been exposed to COVID, and it's more people than we have a positive test for. All right, so someone asked another really great question. Why aren't there vaccines for coronavirus that cause the common cold? There's been all this work for COVID, but the common cold has been around much longer. Why don't we have a vaccine for that yet? So I think there are a lot of different factors that go into this. I'm going to keep myself to just 2 or 3 points, because I could talk about this a lot. One is an issue of priority. The common cold doesn't kill many people. It doesn't cause serious illness. And so as a priority for developing a vaccine, it's not that high a priority. Certain kinds of viral infections like colds do cause death, like influenza, and we do have a vaccine for that. But common cold, not that serious. And so the impetus to develop a vaccine, not as high. The other major reason is that a vaccine that worked against coronavirus would still not be that effective against the common cold, because there are over 200 kinds of viruses that cause the common cold. So a coronavirus vaccine would take out some of them, but for the rest, it probably wouldn't do anything. So things like adenovirus, rhinovirus, parainfluenza, on the order of 200 other kinds of viruses would still cause the common cold. All that said, there is work being done right now in developing a pan-coronavirus vaccine that would be effective at preventing infection and disease, both from COVID with SARS-CoV-2 and other coronaviruses. And so we may actually have at some point a pan-coronavirus vaccine that will prevent against that. We'll still probably have the common cold because of those other kinds of viruses. And then this last question, which again, I'm going to have to keep myself short because I could again talk about this for hours. Outside of COVID, what do you see as the most impactful thing in the world of infectious diseases that's on the horizon for first responders? I know that as first responders, you're always thinking about what's the next crisis, what's the next thing we need to expect, and that is so great. That's why we're prepared, and that's why we've made it through as far as we have. So I think things to think about are both the unknown, so the next viral respiratory pandemic, which is probably not going to be the flu this year, but it might be the flu in a few years from now if we get hit with a particularly bad strain of pandemic influenza. And first responders are always going to be at the front lines of people getting sick and having problems at home. So thinking about viruses and infections that are transmitted by breathing, the easiest thing possible. Those are always going to be things that first responders need to be thinking ahead about. Bloodborne infections. So as first responders, you're also encountering people who are injured, cut, bleeding, and like with COVID, use PPE. With bloodborne infections, you use universal precautions, gloves, making sure that you don't come into contact with fluids. But I do think that we see a lot of hepatitis in the community, and hepatitis is highly transmissible through blood. We're doing a great job in the world of infectious diseases of treating people with hepatitis and curing hepatitis C, but we still don't have a cure for hepatitis B, and that's very transmissible. So thinking about the kinds of vaccines you can use to protect yourself, there is a great vaccine for hepatitis B, and so continuing to protect yourself with against things that we have vaccines for, and doing your best with PPE against things we don't have vaccines for, but we have treatments for, like Hep C. And then HIV is something else where it's not, there's not a huge number of cases in the Seattle, Snohomish area of HIV, but it's out there and it can be transmitted through blood contact. And fortunately, we're making a lot of progress with an HIV vaccine. That's another thing that Julie Sartosky and I spend a lot of time working on is other vaccines. So we have hope for that. Hopefully not a huge issue for first responders, but something that we certainly want to keep thinking about. Then the last, I'll just get my last point in about things for first responders to be thinking about in the infectious disease world is bacterial infections for which we don't have good treatments anymore. So right now we have antibiotics that treat a variety of infections. And I know that first responders, by virtue of your job, you're at risk for traumas, bone injuries, bone breaks that may require surgery. Surgical sites get infected with bacteria. You're at risk for all kinds of trauma, smoke inhalation, which then makes your lungs be at risk for infection on top of that, which can be with bacteria. And again, right now we're pretty good. We've got antibiotics, but there's a lot of antibiotic resistance developing. And that's the thing that keeps me up at night once I've gotten to sleep after a day of, COVID care, is the fact that while our antibiotics are good now, in 20 years, 30 years, I don't know how many antibiotics we're going to have. And so doing our best with responsible prescribing, using of antibiotics, not taking antibiotics for things that they don't work for, and making sure that antibiotic courses are fully completed so that resistance doesn't emerge. So those are my 2 cents on things that I would think of first responders needing to keep a particular eye out. I think first I want to throw it to Julie Sartosky and see if she has anything she wants to add, especially about the immunity after infection and vaccines.
Yeah, thanks, Adrienne. That was fantastic. So that slide that Adrienne was showing earlier, that was going over, those were some of you are in there. So those were folks that were part of our cohort study, and we looked at the first folks that got vaccinated after they had had COVID, and then we compared them to folks that hadn't been vaccinated or hadn't had COVID and got vaccinated. And one thing that was also in that slide, it showed that for folks that had not been vaccinated and had not had COVID, the F there on the, yep, right there. In our lab, we tried to infect those cells with different strains of COVID. And everybody who had not had COVID and had not been vaccinated yet were able to be infected with every strain. And that's really important because, you know, we've heard about this term novel virus, and it means that we don't have any natural defenses. We haven't seen it before, and we're all really vulnerable. And that's really important, and why it's scary for people who are looking at data like this, because if we were to do this with flu, you wouldn't see it so clearly that everybody was able to be infected. Because, you know, flu's been around, we've seen it, some of us have, you know, some level of something there. But that's just one point I wanted to bring up.
Well, I want to thank all of you. I appreciate the attention, the attendance, the questions. And for those of you who have heard us talk about the research and have been interested in participating, we're so grateful for your participation in that, as well as grateful for the services that you provide in our community.